Hi, Charles. Welcome to the podcast.
Thank you. It's great to be with you.
So, I wanted to start with something basic when we think of human health and metabolism. Let's start with the basic question: what is metabolism? How do you look at it?
Okay. So metabolism is the set of all of the reactions that converts everything that we eat into everything that we are and do. So it's how we generate biological energy, ATP, from the foods that we eat. It's how we generate every molecule in our body and repair every molecule in our body. We can't think without metabolism. We can't move without metabolism. We aren't anything living without metabolism.
And interestingly, as you already mentioned about what we eat, so I mean, of course, metabolism, we can think of it in terms of food or like as an organism level. But at cellular level, it is, as you mentioned, a molecule called ATP, right? Which is like energy.
No, it's not just ATP. No, it's so ATP is how we can convert food into energy, but it's not really how we convert food into, cell membrane and nucleic acids. So a lot of metabolism is not fully breaking things down to fully oxidize things and generate ATP. It's to break things down to intermediate components like acetyl-CoA and ribose 5-phosphate in order to make things. Just as important.
Yeah. So, but generally people think of metabolism in terms of ATP, but then there are other metabolic molecules as well, right? As you mentioned, it's ribose, but also a system that you explore, NAD, etc.
Yeah. So the way to think about it is that there's catabolic processes, which is breaking stuff down, which is generally to produce ATP. And then there's anabolic and repair processes, which is to make stuff and to keep it functioning. All of those things require energy inputs, but the anabolic piece doesn't really depend upon ATP production. So I just to bring this in the sort of the realm of cancer biology, there are people that are scratching their heads all the time and saying, gee, why don't cancer cells use oxidative phosphorylation to get all of the potential ATP out of glucose? And the answer is, mainly what a cancer cell is interested in doing is proliferating. So cancer cell wants to duplicate all of its components. So by not converting the glucose all the way to carbon dioxide and ATP, it's able to generate nucleic acids. It's able to generate lipid membrane. It's able to generate polypeptides. And so those anabolic components of metabolism are just as important as the breakdown, the catabolism.
And one of the this anabolic system that you study, it's NAD system, right? Um, so let's kind of define it. What is it, and why it is important in metabolism?
Okay, so metabolism in living things works because of enzymes and coenzymes. So you can generate the same amount of heat by simply burning glucose, with a match. You can generate thermal energy. But as we were just saying, that when living things oxidize fuels to generate biological energy in the form of ATP, the aim is not to generate heat, but to store the energy in a usable form. And so that essentially requires channeling of electrons. And the wiring, so if anybody's ever looked at a diagram of an electric car, there's a gigantic battery, right? And there's metal wires that go to the transmission and that go to the cooling system and the windshield wipers and everything else that moves in the car, the brakes. And there's wires that transmit high energy electrons in order to operate all of the moving parts in a car. Well, there's effectively wiring in all of cells as well. It's not metal wiring. It's largely electrons are transmitted by coenzymes, and the 4 coenzymes that carry the highest energy electrons in the cell are called NAD coenzymes. It stands for nicotinamide adenine dinucleotide. And so NAD picks up high energy electrons. It becomes a coenzyme called NADH. And then NADH donates electrons into the electron transfer chain at the intermitochondrial membrane in order to generate ATP. And in those anabolic processes that we talked about, related coenzyme called NADP picks up electrons, and it uses those electrons to generate lipids, to generate DNA from RNA, and to detoxify free radical species. So NAD coenzymes, you can think of them as the essential components of the wiring of cells. They are the central catalysts of metabolism.
So if we try to simplify it a bit, so all the food gets converted into some sort of precursors, and these precursors, they are directly used in our cells, or they kind of transfer these electrons. And the NAD system, which people just can remember it as a system, carry those electrons to different processes, be it to make parts.
Let's make it clearer.
Yeah.
So biochemistry professors like beginning with glucose because we circulate glucose at high concentrations, millimolar concentrations in the blood. Our alpha cells from our pancreas get activated when the blood glucose gets too low. Our beta cells in the pancreas get activated and make insulin when glucose is too high. So there's a glucose sort of based economy in animals, right? And so glucose can be used as a fuel to generate ATP, and glucose can also be used as a precursor to make nucleic acids. But I'll give you an opportunity to educate all your podcast listeners. How many carbons does glucose have? I'm sure you know.
Yeah, it's C6, so 6 carbons.
Right?
Yeah.
And how many carbons does the ribose or the deoxyribose in RNA or DNA have?
It's pentose, so 5.
5, right? So there's an amazing thing called the pentose phosphate pathway that will take glucose, a glucose metabolite called glucose 6-phosphate, and turn it into a ribose, a 5-carbon compound. And now the skill of an organic chemist to convert, to stereospecifically convert glucose 6-phosphate into ribose 5-phosphate would be, extreme levels of skill. Like that would be really, really, really hard for a synthetic organic chemist to efficiently convert glucose 6-phosphate to ribose 5-phosphate. But essentially, every single cell can do this. Every single cell has a pentose phosphate pathway that can convert the glucose 6-phosphate to the ribose 5-phosphate. And it does so through a series of enzymatic transformations. And there are high energy electrons from the glucose 6-phosphate that are transferred onto NADP to then generate NADPH. And so not only can you get a ribose 5-phosphate from a glucose 6-phosphate, very precisely kicking off a single carbon dioxide in the process, you also get the electrons that basically came from circulating blood glucose. You get 2 electrons put on NADP. And then those 2 electrons that are now NADPH can be used to make lipids. And therefore they can make cell membrane, they can make testosterone, they can make estrogen, and they can also detoxify free radical species. So essentially, NAD coenzymes are electron carriers, electron transmission units that work in conjunction with very specific enzymes to do metabolic transformations without which, no living thing would be alive.